Hello everyone, this section is for Morphology of Virus. After learning this section, you should be able to state the size range of variants, identify the path of a variant and describe their function, distinguish enveloped viruses from non-enveloped viruses, and you should describe the types of capsid symmetry. A complete virus particle is called a variant. Variant morphology has been intensely studied over the past decades because of the importance of viruses and the realization that varium structure is simple enough to be understood in detail. Progress has come from the use of several different techniques, such as electron microscopy, X-ray diffraction, biochemical analysis, and immunology. Although our knowledge is incomplete due to the large number of different viruses, we can discuss the general nature of varium structure. Variants range in size from about 10 nanometer to 400 nanometer in diameter. The smallest are little larger than ribosomes. Virus, mimiviruses, the largest viruses known as variants larger than some of the smallest bacteria and can be seen in the light microscope. Most virus particles must be viewed with electron microscopes. This is the structure of varium. Look at the picture on the left. It is a non-enveloped virus. It is the simplest variant. There is a nuclear capsid. A nuclear capsid is composed of nucleic acid, DNA or RNA, and a capsid. The capsid can protect the nucleic acid. On the right, it is an enveloped virus. It is composed of a nuclear capsid surrounded by a member called an envelope. The envelope usually has viral proteins called spikes inserted into it. This is the structure of variants. Then let's see the capsid symmetry. There are three types of capsid symmetry: helical, exohedral, complex structure. First, let's see the helical capsid. Helical capsid are shaped like hollow tubes with protein walls. Look at the picture. This is a picture for tobacco mosaic virus. It is a well-studied example of helical capsid structure. This is an electron micrograph of the negatively stunned helical capsid. It is magnified about 400,000 times. The variants are usually 15 to 18 nanometer in diameter and 300 nanometer long. Look at the middle picture. The self-assembly of TMV primers into a helical arrangement produces a rigid tube. The capsid includes an RNA genome, which is wound in a spiral and lies within a groove formed by the protein subunits. Not all helical capsids are as rigid as TMV. The genomes of influenza viruses are enclosed in thin, flexible helical capsids that are folded within an envelope. Look at this picture. It is influenza virus Varium. It has an envelope. Influenza viruses have segmented genomes consisting of 7 to 8 different RNA molecules. Each is coated by capsid protein to create 7 to 8 flexible nucleocapsid. The envelope has spikes that project 10 nanometer from the surface at 7 to 8 nanometer intervals. The nucleocapsid of many animal viruses, some plant viruses, and at least one bacterial virus are surrounded by an outer membrane layer called an envelope. Animal virus envelopes usually arise from the plasma or nuclear membranes of the host. Envelope lipids and carbohydrates are therefore acquired from the host. In contrast, envelope proteins are coded for by viral genes and may even project from the envelope surface as spikes, which are also called papillomas. In many cases, spikes are involved in virion attached to the host cell surface. Because spikes differ among viruses, they also can be used to identify some viruses. Then let's see Exohedral Capsid. An exohedron is a regular polyhedron with 20 equilateral triangular faces and 12 vertices. Exohedral capsids are the most efficient way to enclose a space. They are constructed from rune or knob-shaped units called capsules, each usually made of 5 or 6 protomers. Pentamers have 5 protomers. Hexamers possess 6. Pentamers are usually at the vertex of the icosahedron, whereas hexamers generally form its edge and triangular faces. The variants of some RNA viruses have pentamers and hexamers, constructed with only one type of subunit. Look at this picture. This is the Anion Virus. On the left is the electron micrograph of Anion Virus. It has 252 capsomers. On the right is a computer-simulated model of an adenovirus variant. The last is capsid of complex symmetry. Most viruses have either icosahedral or helical capsid, but some viruses do not fit into either category. Poxviruses and large bacteriophages are two important examples. Poxvirus variants are among the largest of the animal viruses, about 400 by 214 by 200 nanometer in size, and can be seen with a light microscopy. They possess an exceptionally complex internal structure with an ovoid to brick-shaped exterior. This picture shows the Variance morphology of a vaccine virus. Its double-strand DNA genome is associated with proteins and contained in the core. a central structure shaped like a biconcave disc and surrounded by a membrane. Two lateral bodies lie between the core and the variance outer envelope. Some large bacterial phages have variants that are even more elaborate than those of poxoviruses. The variants of T2, T4, and T6 phages, we call them T-even phages, that infect E. coli are said to have binary symmetry because they have a head that resembles an icosahedron and a tail that is helical. Look at this picture. This T-even bacterial phages has a capsid head. The icosahedral head is elongated by one or two rows of hexamers in the middle and contains the DNA genome. The tail is composed of a collar joint. Join it to the head. A central hollow tube, a sheath surrounding the tube, and a complex base plate. In T-even phages, the base plate is hexagonal and has a pin and joint tail fiber at each corner. Considerable variation in structure exists among the large bacterial phages, even those infecting a single host. In contrast with the T-even phages, many other phages that infect E. coli have true icosahedral heads. T1, Ts, and lambda phages have sheathless tails that lack a base plate and terminal in rudimentary tail fibers. Coliphages T3 and T7 have short tails without tail fibers. Okay, that's all for this section. Here is a word list for you.